Good afternoon, good morning, everybody. Welcome to an update and an interactive session on question and answers that you may have about research and scleroderma in general. My name is Dinesh Khanna. I'm a rheumatologist. I direct the scleroderma program at University of Michigan. I do clinical research, outcomes research, trying to understand, how to measure changes in treatment and side effects of the medication. And we are one of the largest scleroderma programs in the country that's located in Ann Arbor, Michigan. And I would like to also welcome Professor John Varga, a well-known international expert in scleroderma, and my boss at University of Michigan. So John, would you like to introduce yourself?
Thank you, Dinesh. Hello, everyone, and welcome. I'm John Varga. I'm a physician scientist. I've been long interested in scleroderma. I ran the scleroderma program at Northwestern for a number of years. And about 8 months ago, we relocated to the University of Michigan. And then the main stimulus for relocating here to the University of Michigan is that we felt that we have an unprecedented opportunity to build a really state-of-the-art, cutting-edge translational research program with the aim of better understanding scleroderma and identifying and developing novel treatments and interventions for treating and even potentially preventing complications of the disease. So look forward to this interactive session and look forward to your questions and comments.
So John, our first question is that how can people get involved, people living with scleroderma get involved with our program? And maybe I can start. I think like John mentioned, we are very fortunate to have the critical mass and the infrastructure at Michigan to be able to make key discoveries. And I'll just highlight some of the work that has been done at Michigan before Professor Varga came here. We have made a lot of contributions, including, leading the tocilizumab trial. And most of you know Actemra or tocilizumab is now FDA approved for scleroderma ILD. And most of the work was done at Michigan that led to that FDA approval. Our goals now are to really set up a biorepository, and biorepository is linking the clinical and the biological data, your skin tissue, your blood samples, your urine samples, and other samples together, so we can move towards precision medicine. And what is precision medicine? It is really trying to give the right drug to the right patient. And that's what we are trying to achieve with Professor Varga out here. And of course, you can always contribute and be part of the clinical trials and other research that is happening out here. John, do you want to add to it?
I want to just amplify on what Dr. Khanna said. One of the big realizations in scleroderma in the past decade was the realization that people with scleroderma actually look very, very different. They have different clinical manifestations, different severity of disease, different types of organ involvement, different rates of progression, and most importantly, they react very differently to different treatments. And so this variability among patients with scleroderma is a key insight and will allow us to really better target and tailor therapies to each individual person, depending on the course of their disease, whether they are active, remitting, relapsing, progressive, or stable. And very importantly, the technologies that are now becoming available, such as single-cell RNA-seq and ATAC-seq, and many of these very sophisticated technologies that can take absolutely minuscule amounts of tissue or blood and yet generate vast amounts of information that we can do, and then we can link that to the clinical information, is beginning to give us really unprecedented insight into the variability and the molecular underpinnings of the disease. And then based on this kind of information, and I think Dr. Khanna will explain that a little bit, we are now allowed to really pick rational therapies rather than just randomly picking them. And the expectation is that this kind of treatment approach will lead to both much more effective treatment, earlier treatment, and better treatments without all the side effects and risk of conventional therapies. So I think with the application of these newer technologies, which very much involve you as the participants in the research, we are probably facing some real major breakthroughs in the next 1 to 3, perhaps 5 years going forward in terms of improving and significantly improving outcome of people with scleroderma, and hopefully even preventing some of the serious complications. So Dr. Khanna has a very interesting example of what we're talking about.
Yeah, thank you, Dr. Varga. So I want to highlight and amplify something that Dr. Varga just mentioned. To make successful treatment contributions in scleroderma, there are 3 critical things that you need. You need key investigators who understand fibrosis and scleroderma. You need the infrastructure to be able to do that. And you need the technology that is now available at Michigan, among other centers, to be able to do that. So what we planned and do now at Michigan is, here's a person with scleroderma. We tend to do 3 millimeter skin biopsies, and most of you who have seen me or Dr. Varga have gone through this procedure. But now we are able to separate cells, single cells within the skin tissue, whether it's related to collagen, epidermis, skin, other aspects of skin, into the different aspects. So what you're seeing out here are cells that are fibroblasts, which are the key cells that cause thickening of the collagen, differentiating people who are healthy volunteers without scleroderma versus scleroderma. But like Dr. Varga pointed out, we can do pathway analysis. In other words, link different proteins and cytokines and genes that are crossing with each other and try to pick up medications that may be more appropriate for you versus not. Now, all of this is research for now. So I don't want to raise your hopes saying that you come to Michigan and we'll be able to pick up the right target for you. But like in breast cancer, how things have evolved, you can do a biopsy of the breast tissue and pick up the right medication that may give the most benefit and least toxicity. I think we are getting out there. John, do you want to answer other questions? There's a question regarding, let me look here, regarding research on treatment of reversing skin fibrosis.
I don't see that one. I would say that many of the current treatments, at least the ones that we currently use in the clinic, are really more focused on slowing down or perhaps even stopping the process. For example, if we can quiet down the activated myofibroblasts that are these key cells that Dr. Khanna was just talking about, they will no longer cause damage to the skin. And then if we quiet these cells down in the skin, that will potentially allow your own body to repair the damage. Because I think it's very important to remember that our bodies have tremendous resilience and tremendous ability to repair injury, respond to injury, and ultimately repair a lot of the damage, not all of it, but a lot of it. And so some of the treatments that we'll be looking at in the future are kind of treatments to enhance the resilience of your body, not just get rid of the bad cells or stop the bad processes, but to improve the overall health of your blood vessels, of your skin cells, of your inflammatory cells, so they can actually repair and regenerate the damaged tissue. And this is why early intervention and very targeted intervention will be so critical, so that we can have treatment that increases resilience, improve healing, and this is total holistic healing, not just a particular cell type, but the whole body, and repair and regenerate some of the damage that has occurred. And so with this philosophy of treatment, we are fairly optimistic that although a cure for scleroderma is still quite a ways off, ways to not just slow the disease down, but to allow the body to heal a lot of the damage is definitely, within the horizon.
Thank you, John. There's a question from Jane, and I don't know whether people can ask questions. I think it would be great if people can ask questions. It could be interactive. But the question is, are there molecular biomarkers in a patient that is consistent from one organ type to another? And the answer is yes. I think this is the work, seminal work by Professor Varga, Professor Monique Hinchcliffe, and Whitfield that was published. And I will try to do justice to it and then invite Dr. Varga to expand on it. So one thing that is very interesting in scleroderma is that when a person with early scleroderma comes to you, and let's call it a diffuse scleroderma, diffuse meaning you have quite a bit of skin thickening, outside of phenotypically, people look similar. People have, thickening of the skin, they may have internal organ involvement. But at a molecular level, at a gene expression level, they can't be more distinct. So here's an example. This is a work again done by Professor Varga when he was at Northwestern, and Mike Whitfield, who you heard from, I think, today at the foundation. You can look at the molecular expression and phenotype, and here are different phenotypes that you're looking in a patient who has diffuse scleroderma, and it could be fibrotic. A lot of fibrotic pathways are activated. It could be inflammatory. A lot of inflammatory pathways are activated or not. And the most surprising to me is that about 30% of the patients, when you do skin biopsy, are normal skin biopsies or normal-like skin biopsies. And it matters because it helps us define which treatment may be effective for you. And I'll give you an example. We did a trial with a drug called abatacept or Orencia that is FDA approved for rheumatoid arthritis, juvenile arthritis. And we showed in early scleroderma, patients who had inflammatory gene signature had this beautiful response on skin fibrosis, the question that was asked before by Rhonda, I think, about skin fibrosis, whereas patients who were normal-like or fibrotic pathway did not have much response. So that's our focus at Michigan. Our focus really is that can we develop precision targeted therapies that can lead to better outcome measures? And the other question, Jane, that you asked, yes, the molecular profiling within the skin tissue and your liver, gut, others would be very, very similar. They do change over time. We have seen that over a period of 1 to 2 years as people get better, some of these pathways such as fibrotic and inflammatory may go into a normal-like pattern. And that tells us that the people may be starting to improve over time. Dr. Varga, do you want to add anything to what I just highlighted here?
Yeah, I think you hit the highlights, and as you mentioned, some of this stuff is still in the research realm. So a lot of the work that we're doing and other colleagues are doing, but we're very optimistic that we're learning so much from these research projects that this will be increasingly part of routine clinical care, at least in terms of major academic medical centers where there's specialty interest in scleroderma. So that if you come to the clinic at Michigan or other centers where they specialize in scleroderma, they'll be able to do a much more molecular and more precise diagnosis, and again, have a much more rationally based idea of what kind of treatment you need for how long, how aggressive the treatment ought to be. When do you reach a stage where there is a slowing down of the disease or even a remission so that we can back off the treatment? So these are all outcomes that will be enabled by the kind of research that's going on. Now, this research is quite expensive, and we raise money from the National Institutes of Health, the Department of Defense, the Scleroderma Foundation, private philanthropy, to enable the kind of research that's going on. But again, it generates vast amounts of not just information, but even knowledge and better understanding.
Yeah, absolutely, John. Thank you very much. I think we have been very fortunate, and I think somebody asked in the chat where the funding comes from. Most of our funding comes from NIH, which are the federal dollars. The Scleroderma Foundation has been very kind to me, Dr. Fox, and I must tell you that my career started in 2003 or 2004. Kerry would know more, I should know, but it started with the first grant from Scleroderma Foundation that was given for over 3 years when I was being trained at UCLA. So I think that has been very helpful. But there's a lot of our patients, our partners, like we call them, in fighting the disease, our patients who have supported quite a bit of work out here. I think there's a question that I will try to answer. Rhonda asked again, what happens to skin? So this is a medical slide, but I'll try to clarify this. What happens in early diffuse scleroderma is that the skin starts to thicken. So what you see on the axis out here is the thickening of the skin and number of years after the onset of scleroderma. In early diffuse scleroderma, people tend to have thickening of the skin, and over time they reach a peak and then they start to soften. Why skin is so important? Because it's a reflection of what is happening inside the organ. People tend to get contractures, muscle involvement, lung fibrosis, heart involvement, kidney involvement. And in my mind, the skin goes through 3 different phases. Very early phase where people are diagnosed with rheumatoid arthritis, lupus because of positive ANA. They have the puffiness of the hand, and then they come into this fibrotic phase. They come into this fibrotic phase, which is a classical phase, what we call scleroderma or thick skin. And then they go into this very atrophic phase. In other words, what it means is thinning of the skin. Whatever started the process of thickening of the skin at different time periods for different patients just give up. And the skin starts to soften. But what you can see out here is that this patient of mine is left with hand contractures. They are left with disability. So this is usually the process that happens. And again, what we are trying to understand, Dr. Varga and I, is that why do some people stay thick, have a thick skin for 5-6 years, and some people get thick skin, get worse over 1 to 2 years, and then the skin softens. So those are, again, the precision medicine, the translation of what's happening in the lab and at a molecular level to the clinic. So hope, Jane, that answers the question that you were asking.
There's a question from Wangi, if I'm pronouncing it right, to talk briefly about NIH funding situation about scleroderma-related research. So the National Institutes of Health is actually the largest body in the world that funds biomedical research, and certainly in our country, it's the leading organization for funding scleroderma research. The NIH is divided into different institutes, and traditionally, the institute that's called NIAMS, which is National Institute of Arthritis and Musculoskeletal Diseases, which is headed by Dr. Lindsay Criswell, who's herself actually a scleroderma researcher. They are the primary source of funding for scleroderma research. However, given that scleroderma is truly a multi-system disease, the Lung Institute is also a major source of funding. And what we've been trying to lobby for is to form an interinstitutional unit within the NIH that will allow people from the different institutes within the NIH to come together and say, well, all of our organs involve scleroderma. So the Heart Institute, the Skin Institute, the Lung Institute, the Kidney Institute, and even things like the Aging Institute could potentially all work together. And to some extent, they already do, to help promote scleroderma research and fund scleroderma research. In addition, the Rheumatology Research Foundation, which is part of the American College of Rheumatology, has been an increasingly active source of funding and support for scleroderma research. And interestingly enough, the Department of Defense has been a very strong supporter of scleroderma research. And every year they seem to spend perhaps several $1 million or up to several $1 million to support scleroderma research. So in addition to the Scleroderma Foundation and the Scleroderma Research Foundation, these are the major federal and other agencies that support scleroderma research. And with the new stimulus packages and new investment in recovery and research that is going on at the federal level, we are fairly optimistic that there'll be strong support from the NIH and the Department of Defense to invest in the biomedical research, because it's such a vital part of our economy and our national well-being. And I think there's an increasing appreciation of the importance of that. So we're quite optimistic about that.
Yeah, I would agree. And the other thing I just want to add is, John, I don't know whether you mentioned, is the Department of Defense. The Department of Defense have focused areas of research for scleroderma. Actually, we were awarded, along with Dr. Varga and Mike Whitfield, a large grant on Department of Defense, and they have another call right now, and they're asking for letter of intent. And again, the Scleroderma Foundation, along with Scleroderma Research Foundation, have played a very, very important role in my mind to galvanize the NIH, the senators, your representatives, who really make the decision what are the diseases that should be studied. Oh, yeah, I think that's what she said, that, Wangi 5, that the DoD is giving the Idea Development Award. So, that's exactly what we received. And if you're more interested, you can email us and we'll be happy to talk to you. Dr. Varga, there's a question about, the research. Is the research is being done globally? Are people around the world working together to discover new treatments? Do you want to take that?
That's a very timely question, because one of the interesting things we learned with the COVID is that we can actually interface with and collaborate with colleagues around the world in real time. And so, we don't have to travel to Japan or the UK or South America to meet and collaborate with our colleagues. And with the support of the Scleroderma Foundation, we're just planning to launch a global research network that will meet periodically and bring in investigators from really hopefully around the world to discuss research ideas, current advances, and it will all be done on Zoom. Participation will be all free to anyone who is interested and engaged in scleroderma research. And the hope is that this will really catalyze and accelerate widespread interactions from around the globe to capitalize on the talent that's currently exists in many, many countries, but it's been difficult to coordinate. And again, the Scleroderma Foundation has really taken a lead in supporting and making that endeavor happen. And we actually hope to launch that in September. So that's right.
Yeah, thank you, John. And I think a part of it is what we are trying to understand, the genetics of scleroderma, and to understand the genes that are associated with the severity of the disease or getting the disease. That requires, and that has been going on, multiple countries giving the DNA together in a blinded fashion, so people are not discovered by that, and trying to understand. But all the clinical trials I want to highlight that are going on with the approval of Actemra, with the approval of nintedanib or Ofev for scleroderma ILD, and all the clinical trials, I must state that there are about 5 continents that are participating, from North America to Australia to Asia to South America. All these countries are working together very closely to get enough people in clinical trials. And if you have early disease, or if you have lung fibrosis, Raynaud's, I will strongly encourage you to think about participating in these trials. Dr. Varga, there's a question about the precision medicine for scleroderma only happening at the Scleroderma Centers of Excellence. And I can start with that. That is not true. I think we have the expertise and we have the interest. So to make any discovery, to make any paradigm shift into how you think about a disease, you have to have the critical mass, you have to have the expertise at a center, which we do with Dr. Varga, myself, and we have another 10 people with Susan Murphy, who you probably heard, who does exceptional work with occupational therapy and physical therapy and the clinical care we provide. You have to have the technology, which we have at Michigan, and you have to have people who want to come for clinical care. So all that is required within the critical mass to make new discoveries, but that doesn't mean that the precision medicine is not happening at other centers, but their focus might be liver fibrosis, lung fibrosis, kidney fibrosis that can be translated in people with scleroderma.
Yeah, I mean, it's, we live in an era of connectivity and interaction in real time. And so while our center and a few others might be very, very research intensive, anybody can participate. And in fact, there are a number of networks in scleroderma. Dr. Khanna and Dr. Asasi have been very actively involved in a network called PRESS, and there's a number of others that allows many, many hospitals and many rheumatologists to be part of the research effort and contribute patient information, DNA, samples, to really maximize and spread out the involvement so that, all the researchers anywhere in this country can have the opportunity to be part of it. And that anybody with scleroderma who wishes to be part of the research can really, participate by donating skin biopsy or DNA or serum for these networks. So it's really becoming a very integrated and collaborative process overall.
Yeah, I think just to follow up with that theme, and we'll try to get back to the other questions. It's a very interesting question. How do you recruit new researchers? It feels like that there's so few physicians who are really interested or know scleroderma, and that's absolutely true. I got excited because of Doctors First and Dr. Clements. They're like my fathers, and they have mentored me over the past 15, 20 years. And I think that is why it's challenging for an orphan disease to get people excited, and that's why the centers of excellence such as Michigan, UCLA, Hopkins, Texas are the focus as we try to develop precision medicine and try to find new treatments regarding this disease. Dr. Varga, do you want to take that? What about calcinosis? I know you have a big interest in that and digital ulcers. Are there any new research, clinical trials happening for these difficult symptoms?
Calcinosis is a very unfortunate and very troubling complication of scleroderma. It's, as many of you know so well, it's not life-threatening, but it can be really, really almost disabling and a major source of diminished quality of life, causing pain, infection. And as many of you know, most treatments have really failed. But I think a number of discoveries just in the last 2-3 years might, and these are recent scientific publications, might give us actually a first insight into why and how people with scleroderma develop calcinosis. This research is a bit preliminary, but it's published. But that allows us to really start to validate the research and start thinking about rational therapies. And in fact, there is a potentially very low-risk treatment that is already being used in children who have a bad disease called pseudoxanthoma elasticum. And these children are born with an inherited genetic mutation, but they build up calcium a little bit like people with scleroderma do. And in these children, in a very small number of cases, this novel treatment, which is basically an inorganic salt, was able to lower the calcium level and led to some improvement in the calcium. Now, we don't really know whether people with scleroderma have the same type of calcinosis, and we certainly don't know if this kind of treatment would work in scleroderma, but it certainly gives us a very clear path and a testable hypothesis, and we're actually organizing a group of investigators really actually around the world, because it will involve the United States and some colleagues in Europe to think about what the first clinical trial might look like. So I think there's actually hope in understanding and potentially treating calcinosis, which has been such a tough nut to crack so far.
Thank you, Dr. Varga. I absolutely agree. And I think highlighting the, really the interplay between the researchers, the clinicians, the foundations, including both the foundations in US and of course the patients who donate their time and effort and skin tissue and other biopsies for us to be able to do this. I don't know this answer, so I will give it to you. Is there any research on the correlation between adverse childhood experience and development of scleroderma? I don't know the answer to that. John, do you?
I don't. I think it's a really great question because in other fields there is an emerging idea of adverse childhood, if I understand the question correctly, having a later impact on disease. We at Michigan are very actively involved in recruiting colleagues from psychology, behavioral therapy, investigators of our colleagues who are working in these other areas that have been more difficult to study, like chronic pain, fatigue, and even sleep, because these clearly have a very profound effect on how people cope with their scleroderma and how their bodies gain resilience to help promote tissue repair. So we're taking a very, very broad approach that will start to think about the entire lifespan exposure and experiences. And I'm sure we'll learn a lot from that. So perhaps this is what you're referring to. I think it's an emerging and very exciting field of research in scleroderma.
Thank you. Thank you. And I think there were some questions before, and I found this slide. And we have launched a very large initiative since Dr. Varga has come to Michigan about 9 months ago, which is to really develop the biorepository and what we call deep omics, a deep dive into looking at the skin tissue biopsy, the blood, the serum, and the stool, and really try to look at the interplay between your genes, how the environment may modulate the genes, whether, in utero, when you were a baby, and after that, the study of the interplay between the gene expression, RNA, and your proteins, your stool samples or microbes, and metabolomics. So if we are in Michigan, near Michigan, and this was really launched by our thankful donors who are the patients at University of Michigan. And I think one thing that I want to thank the foundations and the patients is that scleroderma is an orphan disease. It's a rare disease. People who have it, of course, don't think it's a rare disease because it impacts your life quite a bit. But it's a rare disease, and trying to get NIH interested, and somebody asked the question, does NIH give money to centers of excellence? Absolutely not. We would love to have it. So every time we have an idea, like putting together a biorepository, NIH does not fund it. Scleroderma Foundation doesn't fund the ideas that are not hypothesis driven. So we have to put somehow the infrastructure together so we can start to capture the clinical, the skin, the biological data together so we can answer those important questions over time. So I just wanted to show this slide, all the detailed work that is starting to happen. And if you come to University of Michigan, if you live around Ann Arbor, we'll be happy to see you and have you participate in the study.
And this slide also gives a highlight of how integrated and inter and cross-disciplinary modern biomedical research is, because as you can imagine, this type of research involves biochemists, data scientists, behavioral scientists, microbiologists, endocrinologists, geneticists, and one of the challenges, but exciting challenges, is how do you build a team like that where all these different scientists, not to mention different medical specialists, can really coalesce and work as a team. And I think the strong scleroderma programs that are emerging around the country, the hallmark is that we make these kind of teams happen and work together in an integrated way. And so you'll see that at many of the leading scleroderma centers, this type of integration going on.
Yeah, absolutely. I think we have one more question, John. And the question is really, there are so many scleroderma centers, but with the possibility of many centers participating in a project, how is it decided which funds are used to fund it? Is it private funding? Is it government funding? It is foundation. Maybe I can start and you can add to it. So there are different ways to fund an idea. An idea that is funded by Scleroderma Foundation or by NIH or by Department of Defense or other foundations are usually hypothesis driven. So in other words, hypothesis driven would be that you say that these are the genetic differences in healthy people versus scleroderma people, and this is what we believe will happen. But to reach that, and then it is reviewed by a peer-reviewed and external peer-reviewed experts, so Dr. Varga and I review different grants from different centers and look at the quality of the work, the quality of the investigator, and the hypothesis of the project. So that's how NIH, the foundation, and the Department of Defense funds it. But if you really think about, if you want to really make a paradigm shift in the field, you really need to identify and have an infrastructure, what we call biorepository. And what we talked about, our biorepository is really funded by the private donors and the thankful patients at Michigan. Dr. Varga, do you want to add anything to it?
Yeah, I think that's right. The funding sources that are currently available are incredibly valuable, not only to test specific hypotheses or to discover specific new drugs. They're also extremely important in order to enable young investigators or promising physicians or scientists who are thinking about, gee, I might want to study scleroderma, enable them to launch on that path. Because as you probably all know, universities don't give money to investigators to do research. You basically have to find your own source of funding, which is not that difficult for established investigators like Dr. Khanna, but for young people who might have great ideas and great passion and great enthusiasm to do scleroderma research, well, the universities will say, that's great, show us the funding that will support your research. So these private foundations and the NIH are an absolutely vital source, an indispensable source to allow this pipeline. And this pipeline is really critical because if we don't have a steady supply of talented and imaginative physicians and scientists coming in to focus on scleroderma, it would really slow down the progress of the work. So that type of funding is vital. Fortunately, the Scleroderma Foundation and the NIH are very receptive to that, and they appreciate the importance of this pipeline. But as Dr. Khanna is saying, to really do it right, to do it in an integrated and cross-disciplinary manner, you need the big bucks. And that often comes from donations or from institutes. And one of the focuses that we have is to develop strategies where we can get large donations, whether from the government, industry, even the universities sometimes have large amounts of money set aside to build centers that can, on an ongoing basis, not from grant to grant, but on an ongoing stable basis, train young individuals, collect the samples, organize these interdisciplinary teams, and allow them to go forward and continue their research in a sustainable manner. A lot of this research, as you can imagine, will take a long time and a lot of effort before it generates the kind of information we hope it will. So it takes a village. It takes small donations, large gifts, and a sustained investment in the research enterprise to really make it happen.
Yeah, I don't see any other question, Dr. Varga, and I think we are almost at our time. Are there any other questions, Kerry, that we are missing?
Okay, I'm looking. Do the research centers know what's being worked on at other centers to try to avoid duplication? That's really a great question. I think the interconnectedness that Dr. Khanna and I talked about with these live global networking Zoom conferences and webinars, as well as regular face-to-face meetings, really focus that we should all work together. We should all work in a complementary way rather than compete with one another or duplicate the effort. So I think we've made great progress in that regard. And I think the global community, and I really mean global because we work with colleagues in the United Kingdom, Germany, Japan, India, really around the globe, is much more organized and harmonized and to minimize duplication or wasted effort and really focus on the important questions.
But I also want to highlight sometimes duplication is a good thing. So, if there's a new discovery that is made, you just want to make sure that if a discovery is made in India or Japan, you are able to replicate that in people who live in the United States. Like I highlighted, there's a interplay influence of your genes or your genetics, your epigenetics, which is the influence of the environment on your genes. So it's sometimes it's good that we are validating. It's not duplication, but it's a validation of what other people found.
Absolutely, absolutely.
Okay, I think the last question, maybe, John. I know there are many studies available for recently diagnosed folks, but what about people who have scleroderma for 10 or more years, so long-standing scleroderma? And I absolutely agree with whoever is asking the question, because the focus of all the clinical trials, clinical studies we do are usually in early scleroderma or people with diffuse scleroderma. And I think a part of it is that the clinical trials want people who are actively changing. So earlier part of scleroderma tend to have actively changing spleen, lung fibrosis, and internal organ involvement. And the later part of scleroderma has a lot of damage or fibrosis that is left with the disease. There are initiatives that are going on, including, I'm leading an initiative with Dr. Alain Lescott, who's visiting here from France and working with Dr. Varga and myself for the last 8 months, trying to focus in patients who have limited cutaneous scleroderma, because most of the research has been done in diffuse scleroderma. So I absolutely agree with the question and the concern that you are raising, and all I can say is that we are trying, but again, the funding, having availability of funding to focus on research questions that can be answered in a late disease are somewhat difficult. But Dr. Varga talked about calcinosis being one. I'm working on the GI or the gut involvement, the Raynaud's and digital ulcers. There's quite a bit of research that is going on.
Yes, I agree. And it's more difficult to reverse damage than to prevent it, but that's where resilience comes in. So if we can figure out the metabolic and inflammatory components that are associated with this late stage disease and then improve that, perhaps by nutritional and other types of interventions, it's a lot like aging. You can't reverse aging, but you can slow it down. And I think in some way, people with long-standing scleroderma and long-standing damage to the skin or the lung or the vasculature, if we can, get the metabolic situation so that the cells are at their optimal health, just like when you're slowing down aging, the tissue has a better ability to repair itself or partially regenerate some of the damage. And again, in mouse models, that's feasible. So I think it's a potential possibility, but it's a little bit further down the road than the current treatments, which are really to slow the disease down and prevent progression.
Yeah, so with that, I would like to thank, National Scleroderma Foundation, who has been an unbelievable partner, I must say, in success of my career, but more importantly, what it has provided to people with scleroderma. Kerry Conley invited me. I remember very distinctly in 2003, I was a fellow at UCLA, and Dr. First couldn't do a talk on GI involvement. John, this is an interesting story, and I had a 1.5-year daughter who was running around me while I was giving the lecture, because Pooja, my wife, who's a rheumatologist too, for people who don't know, was in training or had a call that day. And I've been involved with the foundation now for 18 years and had the opportunity to be part of every national meeting. So I thank, and if people have questions for me or for Dr. Varga, you can reach out to us and we are happy to answer. If you want to donate your skin tissue, other kind of support, we'll be delighted to have it. Thank you very much.
Yeah, I also want to second what Dr. Khanna said. The Scleroderma Foundation has been incredibly single-minded in focus and in dedication to spread awareness, lobby Congress, lobby NIH, raise increased sensitivity about scleroderma, raise monies, support young investigators. And this is really a great time because there's a new CEO who's particularly energized and dedicated to work across the country and with the NIH and with Congress to further increase funding and awareness. So I think it's a very exciting time for scleroderma research and for people with scleroderma.
Well, thank you everybody and have a wonderful rest of your Sunday, and we'll talk soon. Bye.
Bye-bye.